Today we're going to talk about chemical fields, and I know I assigned some of this material already, so I hope it will be familiar. And I was going to start out by actually, and you could tell me whether you want to do it or not. I was going to maybe do one exercise basically of the kind you just did in class, but we could skip that if you think you're all right with it. And then we'll talk a little bit about chemical fields and how they work in CompuCell, and then do some exercises on chemical fields, doing diffusion, and then we'll start doing secretion and uptake in CompuCell. And if we really have not enough to do in class, we could try to do a little simple tumor simulation, but I don't know whether we'll have time to get to that today. So just coming back to before, so these are more for the record than anything else, reminding you the structure of the Python classes, the steppable class in CompuCell, twitted code snippets, to remember to use those whenever possible. We talked about adding steppables. So far, we haven't really done that much, but we did do it last time when we added mitosis steppable inside of our simulation. And it's hard to remember all of the different things CompuCell has. So again, you have these menus that I can remind you of the syntax for things. List and iterators. And something that you're already familiar with is that if you're going to use the volume and surface steppables to change the values in Python, you have to remember to zero out the contents of the XML. And we did cell growth. We did the cell growth with the target surface tagged to the target volume so that the tension in the cell stayed constant. And we talked about cell division, the concept of the parent and child cells and inheritance from those, and the option of different cleavage planes. And we looked at the mitosis steppable, and it has that structure where we create a list of cells to divide. We have some condition on the rule for cell division, which if that condition is satisfied, we add the cell to the list. Then we run through the list of cells to divide, have the cell divide either randomly or with some fixed orientation. And then we have this update attributes function that in particular typically takes half of the volume from the parent to the child, clone parent to child, copies the properties of the parent to the child. It's possible to not copy everything, although we usually do copy everything. And we also talked about scientific plots, which you've just done. And one of the things that you have to remember in a scientific plot is that whatever handle you give to the plot window, you have to reuse in all of the commands going forward. And also, when you create a data series in the start function, you have to have the same name for the data series when you add this is add plot window, sorry. When you add data to that series. And so in the example that you had, the problem was when you added the data point, you didn't have the name of the series batch. And we also talked a little bit about histograms. That's probably hard to see with the light here, but creating a histogram is basically the same as creating a regular plot. You have a plot window creator. Looks almost the same as that for a histogram. I mean, for a regular data series, then you add a histogram series here, and then when you add the histogram, you're not adding a data point. You have to have a list which contains the items to be histogrammed. And you give it, again, the series name has to match across here, and you can control the number of bins. And by default, every time you replot the histograms, the old one disappears, which is not true for a regular scientific plot. And we define tracking fields. And the key thing about tracking field is that the tracking field is created in the init, just after the steppable base init. And you give it a field name, which will appear in player, and an attribute name, which is a dictionary key. And then if you give cell.dict, the same dictionary key, whatever it is, and assign it a value, then that becomes a plot that's visible in player. It's possible also to have a vector tracking field, in which case you'd see little arrows drawn on each cell instead of a color. Again, the thing that you always have to remember, whether it's a regular field, tracking field, or a scientific plot, is that name that you've given when you did the creation has to be something that you remember to use exactly later on. Now, we talked last time when we were doing cell division, we talked about various things that could affect the growth rate of cells. And we had as homework an example where I asked you to do contact inhibition of proliferation. Were you able to get that to work? Did you do that homework assignment?
At the bottom, I didn't do yeah.
Okay, so if you didn't do it as a homework assignment, why don't we try it in class as a simple problem? So why don't we start out with, do you have code that does cell division already?
Oh, yeah.
Okay, so let's take your code that does cell division already, save the project as a new name so that you have it. Don't clobber your existing code, so give it a new name.
Here, but also increase the growth part. So,
Well, to begin with, let's just start out by, let's change the shape of the domain to be 40 by 400 instead of 200 by 200 or whatever you had.
so it's not a pull-out initializer.
Well, we'll first thing to do is change the size of the whole lattice to be 400 by 20 by 40. And this is actually, I think, an exercise that Pedro developed a few years ago. And so he makes a comment, choose the boundary conditions wisely. So you may want to think about whether you want the X and the Y axis boundary conditions to be fixed or periodic.
I think it needs to be fixed.
Okay, if you want them to be fixed, that's all right.
Fixed or periodic, or
Well, if I imagine this to be, well, I guess it depends what you think is happening. If I really imagine this to be a rigid culture dish, then probably fixed boundary conditions make sense. Certainly at the bottom of it, I would expect to have fixed boundary conditions. And we are not, if we have the bottom fixed, then the top has to be fixed too. The left and the right, I might think would be periodic, but using fixed boundaries is fine if you want. And then instead of putting the cells in the middle, you could use uniform initializer to put the cells at the very bottom, one layer of cells at the very bottom of the tube. And to begin with, you could have them grow at a constant rate and see what happens. And Pedro, you could jump in if you have any comments about this, since it is your exercise.
I think we're doing fine. So
okay.
Yeah, it's supposed to be open-ended a little bit.
Yeah. Well, we have more slides. This is just to get the initial configuration going. So.
The extra.
So whenever you are ready to show something, it's fine. No, there's no rush. Take your time.
Created here and
okay, that looks fine. Did you put some cells at the end? I can't see.
I didn't. I think I put some stuff that will show up. So I think it's here. Radius. Oh, if I'm using.
So I wouldn't use uniform initial. Yeah, uniform initializer is fine, but the command for uniform initializer is not radius.
Yeah.
So if you just select uniform initializer, comment out, and replace is fine. Okay. Okay, there you go. And so I think you box min on X to be all right, 0, and you want to start at 0, 0 probably. And Y 0. And then X would be, I guess, 40 or whatever your size of it. Okay. And you just want one layer probably of cells. That's going to give you a couple of layers.
There's like 4 layers. Yes.
Yeah, but to start with, just make one layer.
Okay, so you need to like
that would be fine. 8 would be fine. Well, that's one cell. That's all right too. One cell is enough. Try that.
Okay, so
okay.
One cell up here. Oh, but let's see grow. Maybe the chart, like the initial volume is too big, so it'll shrink a little bit.
That's okay. Let it run. Just let it run and see.
Now it's like growing and
yeah. Now, do you have a volume constraint or do you have a target? I mean, do you have what's your growth law for this?
Yeah, I have a target
okay,
volume.
So you have a constant. Let's say start with a target volume.
Target selfish. I think it is in this program. So I don't have like target.
That's fine. You don't need a target surface for this exercise.
Okay. Just bullet.
Okay, good. Well, to make your life easier, I might make the growth rate a little faster. So it's
oh, I think.
Oops, why did it disappear on you?
What? Oh, beside 8.
The cell disappeared.
Yeah, it's weird. Let me just rerun it. And I think why it's running so slow is because I'm calculating the pressure. So
yeah, but that should take almost no time. But you could comment that out if you want.
okay. This is rolling. I think that's enough.
Okay, let's see.
Wait. 1 month. Oh,
Oh, it's tricky when you comment out a multi-line.
okay.
If you use the block comment command in the editor, that will do it, those things correctly. So you don't have to do it by hand.
Understand.
So let's look at your step function.
Let's see. Here. Oh,
You have a blank start function now.
so I have to like just remove this.
That would be
error.
okay, or you could put a pass in it. Would be okay too.
Oh, okay. Yeah.
You're not allowed, remember, Python, you're not allowed to have empty blocks.
What is the?
46.
I didn't see that. Sorry.
All right, fine.
But,
Okay.
like, there's,
So your cell is disappearing when it divides. So let's see why that is.
but I don't like see a cell. Do you see cell appearing?
Yeah, it starts and then it disappears.
Oh, okay.
When it divides, it's losing somehow when you're doing the cell division, you're losing the properties of the cell.
Maybe like how get.
Okay, so let's come back to the syntax for cell division. This is why I always review things at the beginning of class from the last week so that we have an opportunity if there's something that isn't quite clear. So when you have a cell divide, you have to give the daughter cells attributes, otherwise they have no attributes. And so there's a function called def update attributes. There you go. There you go. So what happened to that before? Okay.
Now just divide.
So what you have there is correct.
Yeah. Like, yeah.
Okay. Is that the end of the code or is there more below it? No, that's
No, I just delete all the.
That's
like,
fine. That's fine.
attributes.
Okay. So let's run that now again.
Okay. Do you want like run with that?
I just run it.
Okay. Oh yeah.
Okay, so when the cell divides the second time, they're disappearing. So let's see why that could be.
This is the Twitter page.
Okay, so let's see. So okay, if k is greater than 500.
Just like this
So
line requesting target volume to double.
Okay. So what is divide threshold set to?
Like after 500 like hollow steps, and the target volume just doubled.
Right, but in line 66, where is the value of divide threshold set?
It's just like in line 64.
Okay, I see. Cell.dict V0 times K. Okay, I'm not sure. What are you trying to do here?
I think this is a model from like the assignment, and this is like the target threshold is like growing, and so like the threshold that the cells decide to divide is also growing. So I just want to keep like remembering the value.
Okay. Why don't we just do something simpler though for the moment, which is let's just I think what's happening is the cells are dividing a lot. Once they start dividing, they're dividing and dividing and dividing. So why don't we just set why don't you just have this replace line 66 with just if cell dot volume greater than 60, then do it. Make things a little bit simpler. Because I think what's happening is the cell is repeatedly dividing because of that code. Okay. So why don't we try that, see what happens.
I didn't share the. There's no cell a period of as well. Like,
So,
if there's shrink.
okay, so if the cell is shrinking from the beginning, then let's see, look at the beginning. Let's look at the start function.
Even the glowing one? Yeah.
Okay. So where do you define the target lambda? Where do you define lambda volume and target volume?
Here, like in the constant measurements.
Okay. That looks okay. So why is the cell disappearing on you? Do you have a very large volume? Do you have a very large contact energy?
Let's see.
Let's make cell dot lambda volume 10 instead of 3. Let's try that.
Okay.
Okay. And try running that, see what happens.
Oh, now like they just.
There you go, that's better.
use-
Okay. Now you have periodic boundary conditions at the top and the bottom,
like the
which is
previous one.
probably not what you want.
Oh, here. No floods.
So yeah, you don't want no flux. I mean, the default is no flux. If you don't say it's periodic, then it'll be,
There's no floods.
you just delete them, just delete the periodic. You don't need to declare no flux
Oh, okay.
for
Yeah.
the lattice.
Okay.
Now you might want the left and the right sides to be periodic, but not the top and the bottom. So you'll see. Try it. And you'll have to reshare your the player because we can only see
Desktop.
There we go. Okay, so now let's run it. Okay. Yeah, that looks pretty reasonable. Of course, it's basically exponential growth, which is, but that's fine. Okay, good. That seems fine. So now let me grab the screen share back for a second. And you see the cells fill everything in. Seems, of course, in reality, they wouldn't grow exponentially. And so the next thing to do is to plot the pressure. And so we'll use a tracking field to plot the pressure. You may already have it.
Yeah, I've seen it.
And actually here, Pedro suggested plotting the actual cell volume. We can do that too. So you could create 2 tracking fields, one for the pressure and one for the cell volume.
Like 2 windows, 2 separate windows instead, like 2 process environmental.
Well, because for tracking fields, each tracking field is always a separate window. Because it fills the whole, you can only plot one scalar at a time. Because it's a color map.
Oh, okay. I was just like reload this. Reload the code.
So again, it's extra field automatic tracking is where to find it. And remember that the creation of the field goes in the init.
Oh. Oh,
That's actually, this must be a fairly old slide because it's actually okay, this is a very old slide. Don't do it this way. Use the tracking field.
yeah. Okay.
This slide actually uses the syntax we had before we had the automatic tracking. So if you go to so the first thing is put your cursor in the just in the init after the okay, that's fine there. Okay. And go to CC3D Python. Extra fields automatic tracking. Track scalar cell attribute. There we go. And that's what you need. And you'll need two of them, one for volume and one for pressure.
Okay.
Okay, and now in the step function,
I'll just delete this part.
in the step function, in that for cell and cell list, because you need to do it for all cells, you want to copy the value of cell.volume into cell.dict for that.
Do we have to like enter?
So just,
No.
yeah, so you'd say cell.dict and then the name, whatever the key was that you gave it. Pressure would be equal to cell.pressure and cell.dict volume, cell.volume. And you probably want a minus sign in front of pressure because the default is the sign of pressure is backwards.
I'm sorry, like
Pressure is backwards, so you probably want minus cell.pressure.
minus
Minus,
like
no, minus, a minus sign in front of cell.pressure. A minus sign?
yeah, like this one. Like this one.
Multiply cell.pressure by -1.
Oh. I'm sorry. Oh, okay, I see.
It's just cell.pressure is usually a negative number, so you want to multiply by minus. There.
Sorry.
So, okay, try that and let's see what you got. And maybe screen share it again and we can take a look. So you'll have to add a window. To add a window, you click on the to add a window, and I'll go into player. Go into player. Click there. Okay, and now you have to select cell field, you have to select volume and pressure.
Oh, that's how you. Oh, okay. I thought they like automatically pop up. Yeah, this one's for volume.
Okay, now you can run. So, yeah. I don't know why the color scaling isn't working.
Okay, now I think afterwards they'll start working. Oh, it's sort of blue.
So if you look at the volume, if you look, it's a little hard for me to see here in the bottom left-hand side of the field. At the bottom left, it'll give you the minimum and maximum value that are present. What are the values?
The minimum is 12. It's like shifting between 12 and 13. The maximum is 20. Yeah, that's for the value.
Okay.
And this one for the pressure on the minimum value is they're still increasing in the now they're over 200,000.
Okay, so what's happening here that's a problem is that these colors are not auto-scaling. So you must have done something where you fixed the scales for the colors. And the problem with Player is it tends to remember what you did before. So don't typically you don't want to close windows. Just okay. So go to Player. And now go to the settings command for Player. There you go. And now we're going to go to Field. And now you have pressure and unfix the maximum. So, okay, fine. So then hit OK. You have to hit OK. You have to hit OK each time.
Oh, okay. So it's
Have to hit OK.
oh, now like you see.
And now I think, okay, now you can see both. Okay, so now start it over. And now you should be able to see what's happening.
I just need
You've got them both set to pressure.
change to color.
Okay, now hit run. And now you'll see the
That's really reasonable.
so the pressure is highest at the bottom and lower at the top where they can move freely. And you'll see the volume is actually going to be, you'll see these waves of compressed cells or even disappearing cells because you're trying to put material into this simulation when there's no room. So, okay, so good. So this is a good base simulation to work with. And now let's come back to the exercise.
Cool.
And so you've already plotted the pressure, that's fine. And so now let's make the growth rate depend on the pressure. And the simplest one to do is to say that the growth rate is whatever your initial growth rate is, times 1 minus the actual pressure divided by the reference pressure. And again, you have to remember that the pressure is normally negative, so there'll be an extra minus sign. So if you say the pressure, whatever the pressure, cell dot pressure, you need to put a minus sign in front of. So in this case, it would be 1 plus cell dot pressure over reference pressure. And something that you probably want to do before you get too involved with this would be start your simulation again. and look for the typical pressure in the cells when there are only a couple of them.
And like for this window.
Well, but you want to hit stop and then hit run again.
Okay.
And now turn it, display the pressure. Okay, and hit pause. Okay, good, it's already paused. Look at the typical value of pressure. What is the range of pressure? I realize it's small on that screen.
Yeah, I think it's -8, and the maximum is 51.
51.
51.
Okay, so if you picked a value of something like 25, what you want is for the if you make the reference pressure too small, the cells won't grow at all. If you make it very, very large, then the pressure will get to be big and the inhibition won't do very much. So you have to pick something which is, quote, reasonable. So a value of 25 might be reasonable. So if you now multiply in your growth term, you multiply whatever your current growth rate is times 1 minus the cell dot pressure over 25, then you can see what happens. Try that.
FP is our FP here is value. Oh, wait, what is the V?
Sorry?
Oh, what is the V here? The content of it.
I'm sorry, I can't hear you.
What is the VG here? It's like I need to think
V target.
V target. Oh, okay.
I can edit the. I wrote it in shorthand because to save space, but okay, I can. Let me fix that.
Yes. Okay. Yes, the G means the growth, right?
Sorry?
G means the growth.
G is whatever your current growth rate is. So you defined a growth rate. What was the growth rate you have in your simulation?
Oh, it's fixed. So like we need a factor to calibrate.
Right. So you just multiply whatever the current growth rate is by 1 minus cell dot pressure over pressure, reference pressure.
Okay. Yes.
So it's just one line change.
Mhm. Yes. Mhm. Okay, because I counted the growth each multiple steps, so it's like not a plus, it's multiplying the growth factor.
Oh, okay, so why are you having the cells grow?
Because I wanted to look very smoothly instead of like after 500 monocle stops and it just doubled. So the growth curve will look more smooth, like smoother.
Yeah, but that's not, okay, that's an
Right.
interesting thing to try, to have the cells grow exponentially like that.
Yeah,
But I don't know of any biological situation in which that actually happens.
Putting like Lotka-Volta.
Lotka-Volterra equation
Yeah, Boston.
is linear growth. It's constant in time.
No, it's like the population divide dt equals like a constant, so that means like this is exponential growth.
Right, but each individual grows at a linear rate.
Oh, yeah.
This is an agent-based model. So remember, if you have them grow linearly, if you have the cells individually grow linearly, the population of the whole population will grow exponentially.
So, okay.
So
Let me just
you want a plus equals rather than a times equals.
Yeah, I might just change to the plus. So if I use the times, I have to recalculate the.
No, you definitely don't want to have times equals,
Okay.
because that means that the bigger the cell is, the faster it grows. Do you want that?
No.
So if you use times equals, then whatever you're doing, the cell grows faster as it gets bigger.
Okay. That's
Now, I'm trying to think if biologically it ever occurs that the cells grow faster when they get bigger. Usually cells slow down when they get bigger, but I suppose if you had a very flat cell and it was taking in nutrient from its top surface, then the amount of nutrient it took in would be proportional to the area. And then the growth rate would go up as the cell got bigger.
But cell growth does not like only depend on the nutrient.
No,
It
but
depends
I'm
on-
just trying to think if I'm trying to think of a situation in which it would behave the way you had it going.
Okay.
So what I'm saying is I can think of a situation in which that would be right.
Okay.
But for our purposes, I don't think that's what you want for the moment. So maybe before you put in the growth, I'll tell you what, why don't you screen share again? Let's see it run just with a constant growth rate. And then we'll add the modulation back to it.
Oh, what do you mean like constant growth?
Just have plus equals. Okay, so you certainly don't want to be plus equaling target volume, because that would double. Plus equals target volume will double the target volume every time step.
Oh,
So you just want a constant value on the right.
so like maybe 30 over 500.
Okay, that would be all right. I'd put a decimal point, so it's a float. 30.0.
Okay.
Okay. That'll take 500 Monte Carlo steps to double. Maybe not, maybe too slow, but anyway.
It's not.
No, it's actually pretty.
Pressure, yeah.
Okay. All right, fine. So now let's try multiplying that.
That's like
No,
too small.
I'm not sure. What are you trying? I'm not sure what you're trying to do.
Is it? Oh, if you like multiply that. Oh, sorry. Use that parenthesis.
Okay. And now multiply by, you can make it 1.0 1.0 plus cell dot pressure divided by
25.
Okay, if that's the reference, okay, fine. See what happens. If I did it wrong, it's not going to grow at all, or it'll disappear on you, but see what happens. Oops. Yeah. All right. I got it backwards. Let's see.
You will go back to check the reference pressure?
Let's see. Yeah, let's see. Or we need I guess P reference, we need to know what the value of pressure was. So weren't you printing the pressure?
Check. It didn't. Maybe I deleted that.
Okay. You need to know what a typical value of the pressure is,
Okay, so
because otherwise, again, you're not going to be able to tell what the scaling should be.
I'll just delete this part first
Yeah, that's right.
and then print. So
Okay, so the pressure, good. Okay.
really, sorry, like this is
There you are.
now like it's minus like over
Well,
the
they're
value.
large, but they're negative.
Yeah, but like at the beginning, I think the pressure is not really correct.
Okay, so let's do a step. Let's just do a step.
Okay.
Instead of run, let's hit step maybe 10 times. And let's look at what you printed.
It's around like 25 for the absolute value.
Yeah, well, that's pretty big. I mean, it's getting bigger than that. So the problem was that 25 was not big enough. You're going to need a bigger number than 25. Let's try 100 for the scale factor. Because that's going to be the pressure at which growth goes from positive to negative.
So like probably you
That might be right. And again, I always type 1.0 or 100.0 just to make sure that things are floats. Python is these days better about that than it used to be. In the old days, it turned things into integers all the time, which was a problem. Okay, good. Try that.
no, I think it tends to like convert numbers to
Floats.
floats. Yeah.
Yeah, it's safer to be a float than an integer. Okay, good. So now it's growing. Good. And now we should see that the pressure should never get above about 200.
Yeah. At least like
Yeah, I
here.
mean, certainly, should be limited to be around 100, but it can go a little bit above it. And when it gets to the very top, it should stop growing.
Okay, let's see.
Instead of trying to grow more and more and more, it should stop. And you should see that it should saturate for the color should be about 100. There'll be some noise. It's never perfectly done. But yeah. So that's actually working pretty well. If you look at the number of cells, which is in the console in the player, yeah, so it'll fluctuate a tiny bit, but it's basically neither growing nor shrinking much. Okay, so now one can play other games with this. One can have instead of having a linear decrease in the growth rate, you could have a Michaelis decrease in the growth rate or Hill decrease in the growth rate. And that's something you can try at home, but you've got the basic thing working here. All right, so I hope, since you said you had worked on that homework problem and been a little stuck, I hope this will help you with that. I mean, sometimes so far I've been trying to avoid that, but sometimes I'll assign a homework problem in a sense a little bit before I talk about something in class. And then if you get stuck on it, don't fuss about it. But the idea is you try it, And then you know when we cover it in class, you know why we're doing it. So maybe this is a little bit like that, where you played with growth, and now we did an exercise in class to explore how to control growth with pressure. Of course, there are other kinds of contact inhibition also that you might want to play with, but that one is a simple one. It works pretty well. All right. So I want to talk now. Do you want to get do you want to take a break? Do you want to get something?
It's so fine.
Okay,
Like,
fine.
it's just that
Okay.
an hour.
Okay, but if you let me know if you want.
Okay.
Okay, so we're going to move on to concentration fields and diffusion. And of course, a field is already a bit of an abstract concept. For a field, the concept of a field is that you have enough individual particles that within a box, there are many particles, but the concentration, but the density within the box is fairly uniform. And so a field is fundamentally a density concept is a mesoscopic concept. Now, chemical concentration, electrical potential, ionic concentration are all fields. Chemical concentration in biology can be a little bit problematic as a field, because for something like oxygen or ATP, perhaps, then you really do have, within the size of a cell, you could draw many boxes that have many molecules in them. On the other hand, for things like transcription factors or large molecules, where you have very few of them, describing those as fields is a bit of a strange approximation, because there really is no length scale over which the concentration varies continuously. You really should be treating them as point objects, as agents. We don't usually do that, but it's good to be aware that mathematically the convergence conditions that we are assuming are not applying properly. Now, there are plenty of things that do behave like chemical fields, and oxygen, glucose, growth factors typically have fairly uniform concentrations, which vary from place to place within the tissue and within the cell. And so these are things that we typically assume as chemical fields. Once we have a field, we have to think about how those fields change in space and time. Biologically, there are three ways we can have fields change in space and time. One of which is advection, one of which is active transport, and one of which is diffusion. And at the moment, both PhysiCell and CompuCell only allow you to model diffusion. They don't have advection or active transport mechanisms built into them. That's a pretty harsh approximation, because actually within cells, at least, active transport is quite important. And in biological tissues, you have advection by blood in particular, and lymph is pretty important. So advection is when a chemical is carried by the fluid that it's dissolved in, and the fluid is flowing. I think we discussed at one point, maybe we discussed slime molds as an example where that's the case. And active transport is actually where there are pumps that are moving things or carrying them. And both of those things happen in biology quite a bit. Active transport at the level of cells is something we can model, because the cell can carry a chemical concentration with it when the cell moves. You could do that in both CompuCell and PhysiCell. active transport within cells is more problematic. Active transport outside of cells occurs, for example, in the fluid in the lungs, where the cilia of the lung actively move the mucus above the epithelium. And there's was a beautiful series of experiments and computation that were done by Paul Goldbart and Nigel Goldenfeld from from Cambridge University and from University of Arizona, where they looked at bacteria and they put bacteria on a substrate, where the bacteria have active flagella, and they move the fluid above the bacteria in very complex ways. And so in bacterial colonies, active transport can also be quite important. Advection is a classic physics question, and the reason that modeling advection tends to be quite challenging is that in order to model advection properly, you have to model the flow of the fluid properly. And doing a full fluid dynamic solver, especially for the complex fluids that exist in biological organisms, is not something that's easy to do. And so typically within tissue, not in the blood itself, but in strobal tissue, you don't have large-scale fluid flow. You do have things like lymphatic flow, and in some tissues like bone, you can have large-scale fluid flows. But in a typical solid tissue like muscle, most organ systems, except for the blood, you don't have a lot of active fluid flow. When you do have to deal with blood flow or lymphatic flow, then you have to decide what you're going to do about it. And to my knowledge, none of the modeling frameworks so far have that built in. You have to do it by hand. You can couple your modeling framework to another solver. CompuCell does let you do some fluid solver, simple fluid solvers within CompuCell for blood flow, but it's quite expensive. The simulation will run 100 times slower than it would without that. TJ's center model package TissueForge was designed so that you can do high fluid dynamics pretty well. So you can actually do fluid dynamics in that package.
So like in simple fluid, they just leave some derivative equations and to solve them.
Well, so you could in principle, so if you believe that the boundary conditions are fixed, so that the boundaries of your pipe are not moving, and you think that your fluid is behaving like a Newtonian fluid, like a simple fluid, and the fluid velocity is low, so it's Stokes flow, then you can use a partial differential equation solver to determine what the flow would be. And so you could couple your, say, CompuCell to a solver package like Phoenix or FEBio.
So these are just all built-in solvers.
So those are libraries that will solve
PDEs.
PDEs, yes. The thing is that you still have to pass the geometry of the configuration to the solver and then pull back the velocity field and then apply it in CompuCell. And you have to do that in a sense, by hand. I mean, you have to write Python code to do that. A few years ago, we had in our CompuCell course over the summer, we had Jupiter, great name, a student who's at University of British Columbia, UBC, and he wrote a package that coupled some simple glue code that coupled Phoenix to CompuCell, so you could do this kind of thing that way. So it's certainly possible to do it, but it takes work. It's not something it's not a prepackaged function. So for our purposes, we're going to assume that we're going to do diffusion because that's what we have. We have published papers where we've done we've done advective transport in a variety of ways. And if that's needed at some point, I could show you how to do that. But it's, again, the big problem is not that you can't do it, it's just that the simulations run much slower because the solvers for doing the flow are more complicated than the diffusion solvers. And so they run slower. You have, of course, other kinds of active transport. You have things like ion channels, SIM-port and antiport ion channels. You have active pumps at cell membranes. Something that CompuCell generally doesn't handle very well and doesn't even exist as a concept at Physicell is the idea of jump boundary conditions. So where you have a membrane and there is a discontinuity chemical field across the membrane. And actually, there's a new diffusion solver called the finite volume diffusion solver at CompuCell that allows you to implement all of these things. I haven't used it much, and we can try it out. Most people complain about it mainly because it's slow. It allows you to do very fancy things, but it's slow. This kind of ion channel and transport at the membrane has been implemented in the package VCell from University of Connecticut. Michael Bleenoff is now the person who is in charge of that. And it allows you to define very complex ion channel and transport rules across membranes. But historically, the cells in VCell weren't allowed, couldn't move. They were fixed in space. So it was really focusing on a scale, one length scale lower than the one we typically do. Within cells, of course, you have things like vesicles that are actively transported by molecular motors. We're certainly not modeling those, although those could be important on larger scales. And we also have endocytosis and exocytosis, which can be active as well. We're going to tend to simplify all those things and just say there's diffusion. If we had a diffusion solver with drift, and actually diffusion with drift is not that hard to write computationally. The problem is what the direction and amplitude of the drift is. But hey, Pedro, that might be something that you'd like to do, would be to add a drift term to the diffusion solvers.
Yeah, it's something.
If you had a vector field and that applied a drift on top of the diffusion solver, that's actually computationally quite simple. You could even do it in a GPU. And as long as you're not actually having to calculate the vector field on the fly, it's not a bad computation. It doesn't make the diffusion solver very much more complicated. Now, of course, the problem is that in our simulations, the boundaries of things move because if we have a tissue, the boundary of the blood vessel moves in every Monte Carlo step, it's a different position. And so that's where the coupling gets a little bit complicated. If the boundaries are frozen, you have a pipe that doesn't move, then calculating flow in the pipe is pretty easy. But if there's pressure, right? If the pressure in the fluid moves the boundary, and the boundary is applying pressure to the fluid, then coupling the fluid flow to the mechanics gets to be a bit difficult if you don't have them integrated with each other. And so that's really the reason we haven't done it.
So that's just not a simple like piling up three procedures. Is
So the problem is, if you have the position of a boundary and you say, okay, given that boundary and given the pressure on the outside of this fluid, what's the velocity of the fluid at every point? That's a computation that you could do pretty easily. But the problem is, when the boundary of the cells move, they're displacing the fluid. They should be moving the fluid out of the way. And that means they're applying a pressure on the fluid, and the fluid would apply a pressure back. And if you do the fluid solver separately from the mechanics, then those feedbacks are being ignored. And so you can have problems where you don't have conservation of volume in the fluid, for example. Or if the fluid has material that it's advecting with it, you don't have conservation of the material, the amount of material in the fluid. So solving fluid flow in something like the aorta is really a hard problem. Even in capillaries, the problem in capillaries is the fluid flow is very rapid.
in capillary, it's rapid.
Yes, it's very rapid.
Okay. Oh, you mean like compared to other tissue fluid instead of like blood?
Well, we're talking about blood, but so let's think about time scales. When we're doing cell division, our cells are dividing every 12 hours or something like that, right? Okay. And in CompuCell or PhysiCell, the intrinsic time steps that we use to describe the processes are typically a few minutes, a minute, a few minutes per time step. Because nothing interesting at the tissue level is happening on a time scale of less than a minute. Now, when we look at blood flow in a capillary and ask how long does it take for the blood to move one cell diameter, the answer is 1,000th of a second. And so that means that the intrinsic time step, if you're doing the fluid solver, is 1,000th of a second. And the intrinsic time step of everything else is 5 minutes. And so now 5 minutes is 300 seconds times 100. So we have 30,000 times more time steps if we're going to include that time scale of the fluid. Now, it's not quite as bad as that because there's some things you could do to simplify, but fundamentally you're running hundreds or thousands of times slower when you need to include that fluid timescale. So when I say fast, what I'm saying is the typical time, the typical speed of change in the blood is on a time scale that's very fast compared to all of the other processes in the tissue. If that time scale is important, then you're stuck. You have to simulate it. But then your simulations are going to be quite slow. And so that's why we really would rather not have to simulate advection if we can avoid it. And again, we've published papers where we do it. So it's possible, but it's expensive. Now, diffusion, of course, is a passive process driven by entropy. And there are a couple of things that are very important to think about with diffusion. I don't know if you've had a course in biophysics or chemistry where you've thought about, typically you'll see the diffusion constant or the diffusion coefficient.
Yeah, I think I learned some general physics in
Right.
university. I learned fluid dynamics.
Okay, well, if you've learned fluid dynamics, you're way ahead of most people in this department. I used to teach fluid dynamics.
Oh my god.
I mean, my PhD is in fluid dynamics, so.
Oh.
But I actually had fluid dynamics from Chandra Shekhar, who had a Nobel Prize in fluid dynamics. When I was a student, he taught fluid dynamics.
Okay, I studied marine science when like in my undergraduate. So when I learned fluid dynamics in my sophomore year, I thought like I was not going to use this.
Well, it's pretty heavy. Mathematically, fluid dynamics is pretty heavy. The Navier-Stokes equations are quite complicated.
Yeah.
The diffusion equation is much simpler, but there are still a couple of key concepts that I want to go over with the diffusion equation. And so I don't want to make life too hard for you in terms of deriving the diffusion equation. If we think of the concentration in a box, and there is some number of particles in this box, and the sides of the box are dx, dy, and dz, and now we think about the flow, the flux through any boundary of this box is given by the diffusion constant times the gradient of the concentration. That's the diffusion flux. So that means this is simply saying that the flow of material is linearly proportional to the difference in concentration, and the direction is along the gradient of the concentration. And it's important to notice that the diffusion constant has units of distance squared per time. And that's actually quite important. That's the flow across each boundary of my box. The net flow is the integral over all of those surfaces, which is the divergence of that. And so the actual diffusion equation is dc by dt, the concentration, which is a scalar, is minus the gradient, the divergence of the diffusion constant times the gradient times the concentration. And often you'll see this simplified to the diffusion constant times the del squared, the Laplacian of the concentration. Now, if the diffusion constant is the same everywhere, this simplification is correct. If the diffusion constant is spatially varying, then you should use this more complicated equation. CompuCell allows you to have a spatially varying diffusion constant, so you can specify a different diffusion constant at every place in space. However, CompuCell solves this equation, the bottom one, not this one at the top. And so when you have spatially varying diffusion constant, the change in concentration that CompuCell gives you is not quite right.
But like, can you specify? So even like they're not continuous?
So if the diffusion constant is the same everywhere in the system, then I can take the d outside of the derivative, and I get d times del squared c. And this is the equation CompuCell solves. if the diffusion constant varies from place to place, then I should have a term which looks like the divergence of the diffusion constant, as well as this term here. And we don't solve that. If the change in diffusion constant from place to place is slow, is gradual, then it doesn't make any difference. But if the diffusion constant changes abruptly from place to place, then there's a term that's left out that should be there in the solver.
I see.
And you're unlikely to notice that. In other words, when you run simulations, that's unlikely to be the thing that's causing them to diverge from reality. But it's important to know that the diffusion solvers do that. A lot of diffusion solvers do that because adding that extra calculation slows down. Again, it's a question of time. Solving the diffusion, the normal diffusion equation here is very fast. Doing the full diffusion solver is slower, and so people approximate. And so there's this red comment here, which is if the diffusion constant is not the same everywhere, especially if there are things like membranes, or the other thing is that we've written D as if it were a scalar, but D here is multiplying. Gradient of concentration is a vector quantity. D is actually a tensor. It's a 3x3 tensor.
So like dxs, dx1.
Yeah. And if I'm in a simple fluid like water or in the ocean, then that doesn't matter. But if I'm in a structured material, like the fibers of the extracellular matrix, flow along the fiber is easier than perpendicular to the fiber. And so then the diffusion is actually not isotropic. It can move faster in one direction than another. And again, this is not something that we provide. If you go to a fancy solver like Phoenix, you can do all of these things, but we don't implement those. Neither does physics. Yeah. Again, most of the time, this doesn't matter. You can get away with what we're doing without a problem. But it's, again, I'd feel careless if I didn't at least mention you should be aware when we're building models, we make many simplifications. It's important to be making simplifications that we know about and not make them by accident. And so that's why I'm mentioning these things at this point. All right. Okay. So CompuCell has, like PhysiCell, has a field, which is an array of floats. CompuCell also has a vector field object, so you can define basically a lattice which has three elements per lattice site to have a vector orientation. You can't do diffusion with vector fields, but there is a vector field if you want it. CompuCell has a bunch of diffusion solvers. Fundamentally, there are 4 of them that people use. There's the new one, which is the finite volume diffusion solver, which is pretty fancy, and which I have to say I don't know very well. And as I mentioned, FVM, diffusion FVM. There is a steady state diffusion solver, which calculates the long time behavior of the system, which we don't use very often either. But if you have something that diffuses very, very fast, like oxygen, and you want to actually have that be realistic, you can use the steady state diffusion solver. There's a diffusion FE, is the one we use for almost everything. There used to be a GPU version of it, and I don't know whether it works anymore. I'm not sure. Pedro, did we ever get the GPU diffusion solver working again?
I'm not sure. I like it was I remember an email saying it's working, but I didn't test it.
Well, maybe you should try it. Because it really speeds things up a lot if it works.
Mhm.
So we wrote a long time ago, we wrote a GPU diffusion solver. And then the problem is that the GPUs change.
Mhm.
And so then the code you wrote for old GPUs doesn't work anymore. So,
Mhm.
and there weren't enough people using it so you'll have to check. I don't know if we have I don't know how old this slide is. And so I don't know if the GPU works for diffusion solver. If it does, it speeds things up a lot. It's worth a try. And then there's also a reaction diffusion solver, but it has sort of limited capabilities. But if you want to write a reaction diffusion equation for patterning, reaction diffusion equation. Have you ever done reaction diffusion equation? Okay. So if you have multiple chemical fields that couple together in a nonlinear way,
Mhm.
that's called a reaction diffusion equation.
You mean like those kind of like substrates would have reactions? Or they could just be independent with each other?
Well, so usually they're not used to represent the chemical reacting directly, although they could. There's a whole branch of mathematical biology devoted to patterning, spatial patterning due to chemical diffusion. And this goes back to the famous paper of Turing, 1952 paper of Turing, The Chemical Basis of Morphogenesis, where he said that if you had two chemical species that interacted with each other and diffused, diffusion by itself smooths everything out. You get uniform concentration after a long time. However, if you have two chemical fields, both of which diffuse, but which have feedback on each other, you can get stable spatial patterning or you can get traveling wave patterning. And that's called reaction diffusion equation. For relatively complicated kinds of coupling, you can get very elaborate patterns. And there are certain kinds of developmental processes where reaction diffusion seems to be actually what is happening. In particular, the coat patterns of mammals. So the stripes on a zebra, the spot patterns on leopards, the stripes and spots on fish, all of those are governed by reaction diffusion equation. And so there are many textbooks, entire textbooks, written about how you define the equations that give rise to those particular patterns that are observed in those situations. The behavior of the heart can also be described by reaction diffusion equation as a continuum, and actually the brain also can be described at a continuum level by reaction diffusion equation. So the concept of coupled fields, coupled diffusion fields with nonlinear interaction is quite powerful. A lot of those fancier ones require more kinds of coupling than we have, but CompuCell does provide basic reaction diffusion equation. There's some demos in Player that show, I think, Schnackenberg equation, Fitzhugh-Nagumo equation, and a few other basic reaction diffusion forms.
I think I can check it to after
Yeah, it's not, again, for today, it's not something you need, but it's, if you haven't run into reaction diffusion, there's a whole, I mean, they're Geiger, Meinhardt, they wrote a series of textbooks on this. Philip Maney has written textbooks on it. There are many very nice. The patterns are quite impressive, and the mathematics is also quite beautiful. So let's just talk about now the computing side of things for a minute. The chemical field initializers in Wizard have changed a little bit since we did this one here. But so far, when we've been doing simulations with Wizard, we haven't created a chemical field together. So we've just jumped over the box that said chemical field. Here we'd say, okay, we're going to do a chemical field. We'll put in the name of the chemical field. We'll hit add. And then we have to select the diffusion solver. And typically, we're going to select diffusion solver FE, although they're going to be the possibility of steady state, reaction diffusion, and now this finite volume one. And we might have multiple fields. We're probably not going to have 40 fields the way you do in your simulation, as that would run pretty slow. Paul solves the problem by basically solving everything with the same time step and everything in his code. We solve each field independently. So it's as you add more fields, it gets slower. And then you have to give the field a name. I'm sorry. And it will create XML. And the XML has a bunch of different options in it, some of which are pretty obvious and some of which are less obvious. First will be the name of the solver, and then the name of the field. And then the name of the field is repeated here. And I don't know what happens if you accidentally make those two things different. Don't. I'm not sure why the field name is given twice. I don't know whether you can do one or the other, but it instantiates with both. There will be a global diffusion constant and a global decay constant.
Okay.
And sorry, go ahead.
Decay.
Right. So we haven't shown decay yet in our diffusion equation, so we're not going to talk about that one. Okay. And then there's also initial concentration. We can define simple arithmetic forms of X, Y, and Z for the concentration, for the initial concentration. And we can also read the initial concentration from a file if we want to. And that's really all we need. Now, there are a bunch of other options that we'll come to in a minute for the diffusion solver, but basically all we really need for the diffusion solver is diffusion solver FE, diffusion field name, and then global diffusion constant. And we don't need any of the other stuff. That's all we need. Something we always have to think about, which is quite important for diffusion in particular, we've already talked about it for the cell lattice, is boundary conditions. Typically, we imagine our simulation to be a box inside a bigger universe. And so we have to decide what happens to chemical at the boundaries of our box, just the way we have to decide what happens to cells at the boundary of our box. Quite often, we will use periodic boundary conditions. So a chemical that leaves here shows up and comes back on the other side. Fairly often, we'll use the equivalent of no flux boundary conditions. Typically, when we do that, we'll say constant flux equals 0. But CompuCell allows you also to define a constant non-zero flux, which would be equivalent to pouring chemical in from the edge, a constant source. And it also allows you to do constant value boundary conditions. The most common constant value boundary condition would be saying 0, which would be absorbing boundary conditions. But if we imagine that, for example, our tissue had oxygen in it, with a constant level of oxygen, we might say that there's a constant value at the boundary, which would represent a source of the chemical. Okay?
Mhm.
And so you do have to be careful about what those boundary conditions do. Here we have a single cell that's secreting a chemical. And in a periodic boundary condition, when the chemical gets to the edge, it comes around and comes in from the outside. Here we have constant derivative boundary conditions with the derivative of 0. That's no flux, which means that we basically have a rigid wall that reflects the chemical here. And you'll notice the concentration is quite high then at the boundaries. Here we've set a constant value of 0, which is absorbing the chemical at the boundaries, and you'll see the concentration goes down. So even though our source here is the same, the field is quite different for the 3 boundary conditions. And so it means that it's pretty important to remember which boundary condition you want to use, because if you have the wrong boundary condition, you'll get something that doesn't make sense. And the default, I don't know whether that's ever been fixed. The initial default boundary conditions for the diffusion solvers, when you used Wizard, had different boundary conditions for the left and the right and the top and the bottom, just to show what they looked like. But people wouldn't change it, and then they'd run it and they'd get things that looked very strange. So you do have to remember the boundary conditions and effects. So within the diffusion solver, there's a block called boundary conditions. For each axis, X, Y, and Z, you could specify periodic or constant derivative at the minimum side, constant derivative at the maximum side, constant value at the minimum side, constant value at the maximum side. They don't have to be the same at min and max. Periodic, you're doing both, but for constant derivative or constant value, you could have constant derivative on one side and constant value on the other.
Okay.
And so again, the default boundary conditions that Wizard gives you don't make sense. You're always going to have to change. I think now that Wizard lets you tick box the selections. We fixed it, the old ones. Okay. So let's create a chemical field with Wizard. And we're going to say there's a global diffusion constant of 1, no decay. We're going to say a uniform initial concentration using initial concentration expression, just the number 1. And then we'll have periodic boundary conditions in X and Y. And we'll run it. And maybe I should be doing the same thing, because I should be looking at how Wizard behaves right now.
No change for the lattice dimension.
Yeah, none of the other things we're going to do, because we're not going to be using cells right at the moment. You can create one cell type because we'll need it in a few minutes, but so I'm going to try doing it myself. So I think that the diffusions. Okay. Yeah, so the diffusion solver has been moved in Wizard. It's now after rather than before the other stuff, the cell definition. So let's see, oxygen. We'll call the cell chemical field oxygen. And we have a choice of diffusion solver, Fe, reaction diffusion solver. Oh, I didn't know there's a finite volume reaction diffusion solver. That's new. Steady state diffusion solver, flexible diffusion solver, fast diffusion solver 2D, kernel diffusion solver. Okay, so we'll just use diffusion solver Fe.
Yeah, keep that. Mm,
And now we create that. Boy, this has changed a lot.
Yep.
Okay. So uh, so you have the first line is the diffusion coefficient, which is the global one. One is, well, it says 0.01 is fine. It doesn't matter what the value is. We want the decay coefficient to be 0. So in the second one, we want the decay coefficient to be 0.
Even for global,
Yes.
so what is the global here mean? Like, do we have any other agent or like objects?
No, well, so we have to say what the default diffusion and decay constants are. What CompuCell will do is you could say within a cell of a given type, the diffusion constant is different. So you can define a diffusion and decay constant by cell type.
Mhm.
In that case, any region that has that cell in it will have a different constant.
Mhm. Okay.
And we're not going to use all these secretion options that are in that.
Okay.
In fact, typically we're going to do what it says here, which is it's by default, if I scroll through this, when I created a cell, it's created a decay coefficient for the cell type that's different from the global one. So I want to make that the same as the global one. So if you scroll down where it says cell, yeah, make that the same as the global one, whatever it was.
Okay, I'll just make it.
Yeah, there you go. Okay. And there's this comment, control secretion through Python. We're not doing any secretion at the moment. You have the initial condition. We're going to use initial condition is just going to be 1.0 for the moment. Okay, and now we have to define the boundary conditions. And I've said to make them periodic.
Oh, we cover boundary. Yeah. But now I can't change. Oh, because it's periodic. Okay.
So
So we can move to.
so that should be okay. Let's see. Okay, now try that. Seems to be running, but where did my player window go? There it is. All right, it's player. So let me move player over here. So here's what I got.
think I've always seen it with this one, like.
So let me see. So the initial, let me start this again. Oops, screen sharing stopped. I need to share. I forget now with player, Pedro, you have to share the desktop, not player, because player closes every time you restart. So, okay. So this is the code. Initial value is 1 everywhere. The cells don't do anything.
Yeah.
And so, and I defined periodic boundary conditions. So the concentration is constant, so there's nothing happens. When I run this, nothing happens. Let's see what you got.
Oh, I think the cells behavior is a little bit different, but
Do you want to share it? Let's take a look.
yeah, okay. So it's, let me just restart it.
Okay. So let's look at the chemical field. Let's look at the oxygen field.
I think the logic bar is a little bit different, but it's.
Okay, so something is funny about the scale bars. Okay, so the value is just 1 everywhere. I can't see again, if you look at the bottom, at the very small numbers at the bottom, what are the values? They're 1? They're
Yeah.
both 1?
Yeah.
Okay, so the chemical concentration is 1 everywhere. So this is not very exciting. It's doing what it should do. You start with a chemical concentration of 1, and it stays 1. All right. So now change the boundary conditions to be constant value of 0 on X and Y. What do you think is going to happen when you do that? Before you do it, tell me what you think is going to happen.
Still like keep them as periodic.
Now we're going to switch from periodic to
Oh,
constant value boundary conditions.
So you change the maximum min to 1.0. Okay, what change do you make? Are you going to fit? I'm sorry. So what change are you going to make here?
You're going to change from periodic to constant value 0.
Constant value 0.
What's that going to do?
So it's initially it will initialize you with 1.0, but like because like there is a sort of sink to absorb all of the substrates, so you just so like the boundaries will be like 0, and as the substrates that you're using, they'll just go to 0.
Yep, that's right. Hey, by the way, Pedro, if you're there, I noticed if I go into the XML now and I say I want to replace the diffusion solver, it says, do I want to comment out the diffusion solver that's there now?
Yes.
It crashes.
Is that a new version problem? Because it doesn't do like that on mine, I think.
Twit allows you to overwrite the diffusion solver.
Yes.
It pops up the wizard window to correctly.
Let me try here.
Because I was trying to edit my XML. I tried, it says, do you want to comment out and wizard the
Yes.
It crashes, it crashed Twit for me. And of course we have that new Twit window which Bart wrote. So So you shouldn't have to start from scratch.
I tried to like change files because like the periodical things and the.
No, but so what are you trying to do? Tell me.
Oh, I just want to change the periodic to.
Right, but
Not-
that's in the diffusion field.
Okay,
It doesn't change anything else,
so
only in the diffusion.
onto all the operations. Try to.
The cell field, the periodicity of the cell field and the chemical field are different.
Oh, okay, sorry.
No, I mean, I should have said that probably. It's not obvious, so good point.
Okay, I'll just replace it with the periodic form.
No, but it does, you're changing them. It's not, okay, well, that doesn't matter. That one is up to you.
Okay.
Because you're not using but what you're doing here is you're changing the periodicity of the,
Periodic.
scroll down where it says boundary conditions. It's those.
Okay. So how you like?
Now, this is what I was just saying. In principle, if you click on CC3DML and you go to stepables and you select Diffusion Solver FE, it should say, and if you say replace, it should.
Okay.
Yeah, no, on hers too, it crashes.
It's because we didn't.
On mine it works fine, but I'm using the old version.
Yeah, so there's a bug in the new version.
Do you just stop the like? You pause the word stop.
Yeah, but it's not a player issue, it's a twitted issue. So let's try this one more time. Let's reopen the twitted. This is brand new, so unfortunately, these things. So we need to open the code you just wrote.
Oh, I usually like start from the beginning.
Well, so,
Just.
but you don't have, if you have player running, you don't have to do any of that. Just go to player.
Oh, but I closed.
Okay.
Player, sorry.
Okay. All right.
Sorry, since I have
There's
to
nothing
start
wrong. There's nothing to apologize for. It's just a little slower to do it that way. Okay. Okay. So now you have to open the last simulation. Okay. And now we need to go to the XML. And I am not sure what we're going to do about this because if it, so you need to, it's the diffusion field that we need. So click somewhere on the diffusion field. Okay, anywhere should be all right. And now go back to CC3DML, stepables, diffusion FE, and replace doesn't work. So let's try comment out and add and see if that crashes too. It crashes too. Comment out and add crashes too. So we can't, so we don't have the, we don't have the GUI for changing the thing you've already done. So let's just do it by hand. It's not that bad. So open it again. Go to the XML. We find where the boundary conditions are defined there, where it says plain boundary conditions. And now we have to type what you see on the screen. Constant value, plain position equals min, value equals 0. So Pedro, what version are you running of Twitted?
4.4.
Ours is also 4.4.
Yeah, I'm a few years behind.
This is 4.7. 4.7 revision 5. That's funny. I don't think I remember the joke in the about in the about comment for the twit. Do you remember that, Pedro?
No, what's the joke?
If you go to about Twit, it says Twit ++5 editor is a free open source programmer's editor originally meant to be an editor for Twitter with limited number of characters to 144. However, after feedback from users, we were surprised to learn that people need more than 144 characters to write software, so we have removed the limitation on the number of characters. As a courtesy to our users, no code written in this editor is cataloged by Google or any other data mining company. All right.
Yeah.
Okay, so now you see, yeah, you see that,
To
right.
be replaced by 0.
right, that's right. So that's absorbing boundary conditions there. All right. Now you could also have, let's do one more. Let's make the top boundary have a constant value, which is 2 instead of 1, a 0.
Okay, so
So now you'll have a source on one side and absorbing on 2 other sides.
source on one side. But like the bottom is also like the
Well, at the moment, all 4 boundaries are absorbing, right? You said 0 on all 4.
Mhm.
Make one of them not 0, but something like 2 or 3, something bigger than the initial value.
so like 3 sinks, 1 source.
Right. And let's make the diffusion constant 1 instead of 0.01, because 0.01 is very slow.
So the diffusion here
Yeah,
global.
right. Make that 1.0.
Oh, I think I have to change the my cell one. Wait, oxygen.
Sorry?
Oh,
Make that 1.0.
okay.
And you can either get rid of the my cell one entirely or make that 1.0 as well. Okay, good. Okay, save. There you go. So now you can see chemicals coming in from the top and leaving on the sides.
That's kind of like what I want to simulate in a gut lumen. But I think like that the simulation result doesn't look quite like you can see that one's
Now, at the moment, your cells aren't responding to that chemical. They're not secreting or absorbing at all, but I don't know whether we'll get to it today, but in my lecture for today, I had planned that we would show you how to secrete and absorb chemicals as well.
okay. It's trying to
If we don't do it today, we'll do it next week. But I mean, but it's okay. So you see,
stable.
it takes a while to stabilize, but it does eventually stabilize.
So
All right. So we played a little bit of some different. So one of the things that I had here as an exercise was to change the initial condition. As I mentioned, you could have simple arithmetic and mathematical functional forms of the coordinates in the initial condition. So this rather ugly thing, max of 0, 10 minus x minus 100 squared minus y minus 100 squared, basically draws a circle of chemical. That draws a circle of chemical at the beginning. So that's something we could do.
This is a square.
Yeah, this is a square. And I don't know whether we really need to do all of these. Another thing that's quite useful to do, which is close to the one that you did here, would be suppose that we had periodic boundary conditions on the left and the right. Why don't you make the sides of your simulation periodic now? Leave the top and the top not 3 and the bottom 1, but make the go back to periodic in the other direction. Why don't you do that and then run that one?
Okay. Thanks. Yeah, so I have to change these. It's periodic.
So put it back to periodic and and Pedro, if you could send an error note to Maciek about the Twitter, that would be good. Okay. So now you're going to get what we call eventually a linear gradient. It's going to take quite a while because you have a big simulation. But eventually it will be it'll be a uniform gradient from top to bottom. Now, I think we could skip this. Let's try one other thing though. We haven't talked about a decay coefficient yet. What if we have a decay? Actually, this one is like getting rid of the decay. Sorry about that. We'll talk about the decay in a second. The defaults in Wizard are slightly different here from the ones we had. So this is basically just showing all the code that we've just done. And the display settings. We've looked at the display settings a little bit in Player. You click on the settings menu and then you go to fields. And the big thing that you typically have to decide about is, do you want constant top or bottom values, or do you want auto scaling of the colors? If you auto scale the colors, then red in one time may not mean the same thing as red at another time. On the other hand, if you fix the top and the bottom values and the values change a lot, then you could get all red or all blue because you're outside of the range of the chemical that you defined. And so you have to play with that. The other thing that you sometimes like is convenient here, which is a bit hard to see, is in the settings there are contour lines that are available to you in player.
Let's see if they still.
And so, yeah, so here where it says scalar contours,
Okay.
and now you have to say how many of them you want.
How many?
So it says nice number of not number where it says number over range there. This has changed a little bit too. When you just hover over it, it'll tell you what it's doing. Move over it. Move.
This one?
No. You had it and then you move there.
Oh.
Number of contour lines, ISO values will be shown as an automatically. So it will draw contour lines, and it'll do that in 2D or 3D. For a lot of things, the contour lines aren't that useful, but sometimes they're quite helpful. So
I'll have to like rerun this.
Okay, yeah, that's pretty much uniform now. Yeah. Okay. So you've got the settings done. Yours is rotated 90 degrees from the one I have here, but you got. I don't think I want to take the time to do this exercise, but I just want to walk you through it. It used to be that when you inside of the Python, when you were going to read the chemical fields, you had to do a get to get access to the chemical field. And so there was a rather complex syntax. That's been simplified. And so if you just type hey Pedro, which of these is right?
Which one is right? Sorry.
There's a typo in one of these, there's a typo in
In her screen. You mean?
and on okay. Yeah, so it's hold on a second. I've got it's in, let me fix it because let me see.
Wait, oh, you mean field and field?
Yeah,
Oh.
I'm pretty sure it's field, not fields. I mean, I'm just sorry. Let me double check. Chemical field manipulation, get field reference. Get field reference. Yeah, field singular. Good. Let me fix that. I don't want to go past a typo without fixing it. Okay. So it used to be that you had to do a fairly complicated thing to read chemical fields. Now the fields are available as Python objects. And so self.field.oxygen is the chemical field.
So it's like an array.
It behaves like an array, but it's not quite an array. It's a swinged object because it's a C++ object.
Oh,
And so there's some things that you can't do.
okay.
So some Python iterators won't work on it. Some slicing won't work on it. But in general, if you say something like field equals self.field oxygen, that will work. And references work. So if I want to read the value at a given location, square bracket 10 comma 10 comma 0 will give me the value at that position. And I can read through, if I want to plot the value across some axis, I could do, this is what you were doing for the pressure, right? I'm iterating over the X range and then reading the value and plotting. I don't think it's worth going through and doing this by hand, but but you could read and write values in the chemical field by hand, by coordinate without doing anything fancy. So in general, the field will behave like an array. The thing I'm going to warn you about is that it's not quite an array, and so there occasionally will be, if you try to do something fancy, it may not work. You may get something like array, the object such and such is not iterable if you try to do something fancy. But as far as reading and writing individual values in the field go, there's no problem. And a standard iterator will work too. In other words, I could have, if I put X colon equals square bracket 0 to 100, that'll work. The standard operator operator iterators in the array arguments will work. And I'm trying to remember what it is specifically that one does that doesn't work. I think it's if you try to do something like so if you tried to do something like np.max parenthesis field, I don't think that will work.
No.
So if you try to get the maximum value in an array,
Mhm.
that's a NumPy function called np max, NumPy max.
You don't have to use, oh wait, oh, it's a array, it's not a list, okay. Okay,
Right.
yeah, you're correct.
And so some of those things work and some don't, and I don't know. You'll have to try it out. I don't know which ones work. Now, for some of those, we have built-in functions. In other words, there are functions that will return the maximum and minimum concentrations of the field and the average. So we have built-in functions to do that, but it's not a NumPy array. It's not a simple NumPy array.
Okay.
And so not all NumPy functions will work on it because it's actually a C++ object presented as if it were an array. So maybe I can give this as a little homework assignment for this week will be this exercise where I was plotting the value as a function of time as a function of position. So there are functions like dot max and dot min on the array on the field will give you the maximum and minimum value. So those exist. There's some other ones that are also there that you can look up. I'd have to look up what all the options are. But that's a case where you can do a dir on the object and it'll tell you what you could do to it, right?
Mm-hmm.
That's easy. Okay, I want to talk a little bit of physics here. Because typically when we talk about diffusion, we talk about diffusion constants and we talk about decay constants. But actually, the things that matter biologically and the things that we really want to replicate in a simulation to agree with the experiment are not the diffusion constant and the decay constant. In fact, very often we will use diffusion constant and decay constants that are quite far from those in biology, because those numbers by themselves don't matter. What matters biologically are the diffusion time and the diffusion length. And so what is the diffusion time? The diffusion time is the typical time it takes a molecule to go a distance, L. And that time is L squared over D, where D is the diffusion constant. And so that means that if I double the distance, it takes 4 times as long to go that distance. And when we say diffusion only works over short distances, that's what we're talking about. Diffusion slows down as we go over long distances. And that makes a huge difference. If I take the potassium ion in water, diffusion constant is 2,000 microns squared per second. For GFP, so small molecule, in E. coli, so that's in cytoplasm, not in water, the diffusion constant is typically 7 microns squared per second. And that number around 1 to 10 microns squared per second is typical for proteins inside of a cell. So again, small ions diffuse 1,000 times, 500 or 1,000 times faster than typical protein. DNA, of course, is a big molecule. It has a diffusion constant it's hard to know what a diffusion constant means for something that's not a point particle, right? If you think of DNA, the moving parts of it move independently of each other. But as a whole, it has a diffusion constant of 510 to the minus fourth micron squared per second. So now we can say, well, given these diffusion constants, and oxygen is comparable to potassium, how long does it take to go some distance? If we are talking about, let's suppose the diffusion constant is 2000 microns squared per second. How long does it take to go 100 microns?
5 seconds.
So 100 microns, right, would be 100 squared is 10,000 divided by 2,000 is 5 seconds. So oxygen takes on the order of 5 seconds to go 100 microns. To go 200 microns, it would take 20 seconds. To go 400 microns, it would take 80 seconds. That's getting slow. And so there's also uptake, which reduces the diffusion length even more. But this partly sets the length scale of the Kaplan number. If we think of a diffusion constant, say, of 100 microns per second, it's faster than GFP. Tens of microns is the most we're going to get to diffuse. So to diffuse a meter would take 300 years. So if you think about a neuron in the body, a motor neuron that's a meter long, if the only thing that were happening in the motor neuron were diffusion, the signal would take 300 years to get from one end to the other. So you have to have active transport, or even more. So diffusion really is a slow thing. And I have another set of exercises here that maybe we can talk about, but not take the time to do, because I would like to at least introduce the idea of how to do secretion and absorption, because I know that's of interest. So one thing that happens inside of cells is that we're not in a fluid like water. We're at a medium which has a lot of large molecules. And so we have what's called hindered diffusion, where we get diffusion around large molecules. There's also what's called steric repulsion or depletion force. It's an entropic effect we're not going to talk about. And that could slow down the effective diffusion constant. We've also talked about how at membranes, what on earth is it? I don't know why it's, sorry, no idea. We've talked about how at membranes, Microsoft wants to advertise to us, I guess. At membranes, we have both hindered diffusion across the membrane, jump boundary conditions, and then we also have things like ion channels that can either be passive, or we can actually have pumps that actively move chemicals. And one exercise, again, I think maybe this is more fun for homework. We can go over the methods, and then if we have time, we can come back to this, and if not, do it as a homework, which is to create a little membrane. And we'll do this with a frozen cell type. And this is not really very practical for doing lots of cells. You can do a few cells this way. Because the thinnest thing we can draw in CompuCell is one lattice site. Then the quote membrane that we can build is at least one lattice site across. Well, in reality, membranes are very, very thin. And so if we're going to make a membrane that's thin compared to the size of the cell, we have to make the cell have a lot of lattice sites. but so that our cells are big and we can't have too many cells in the simulation. But it's something that we can do if we want to. And this set of simulation here, we can actually build a little bit of a membrane by using that option, which is where we say that to begin with here, actually you could do this in your simulation. So instead of making your cells, go back to your simulation and let's make the cells, instead of making the cells, make a line of cells from the top to the bottom of the simulation. So use uniform initializer to draw a bar of cells from the bottom to the top instead of a square in the middle. And actually, wait a sec, because you have the top and the bottom boundaries different. Make it draw a bar of cells left to right instead of top to bottom. So go from x equals 0 to x equals 100 or whatever your top size is.
Decrease the y's. Let's see. Oh, I should also adjust the temperature word length. Oh,
I might make the simulation a little smaller because it'll run a lot faster. So make it 100 by 100.
Okay. I'm gonna go back to change.
Yeah, you'll have to change the initial condition.
Okay. I need to add plugins to.
So the thing that we want to do here is we're actually going to make oops, I'm hitting the wrong button. We're going to make the cells frozen.
Yeah. So,
And I'm trying to, I have to remember what the flag is for frozen cell type. I think it's /freeze. And when you go to cell type, you just would add, it's under cell type. Cell type.
oh, here.
Yeah. So why don't you try, let's see if the Twitter will work this time. Go to CC3DML, plugins, cell type. Let's do comment out of man. Okay, and now retype your cell types. Keep the same ones you had before, whatever they were.
Is it myself?
Fine, but click freeze.
Oh, I see.
Add. Okay, that's fine. Okay, so now you're going to need to fine. So now you know what the syntax is. So it's just freeze equals quote, quote.
Well, like don't you think like like the structure looks kind of like
Yeah,
messy?
it made a mess of the structure. So just copy the freeze equals quote. Just select the freeze equals quote. Just the freeze equals quote. Just copy that. And now undo the change.
Okay.
You'll have to do it twice, probably. Control Z.
Okay.
There we go. Now you can add. Now you can add the freeze. I think it goes with. Okay, fine. Okay, right. You need a space.
Also, kind of remove words, but previous. Do I have to remove it?
No,
Okay, so that's fine.
I think so. The order may be wrong, but you'll see. Try running it.
Okay.
Save it. All right.
Okay. Wait, oh,
Where are your cells? They're at the top.
Here,
You need to
okay.
put them in the middle.
oh, we want to put them in the middle, okay. This should be like
50, 46, that's fine. Fine. No,
No,
you
you
want
want the
100, no,
average,
that's
yeah.
good. Okay, good. Now, at the moment, those cells aren't doing anything, right? They're not
Mhm.
changing the chemical field at all.
Mhm.
So now, what we can do is we can change the diffusion constant in the cells. And this is going to be a case where actually having that plot of the chemical concentration would help us a little bit. Suppose we make the diffusion constant coefficient in the cells to be 0. What's going to happen?
Um,
What do you think is going to happen?
or if the cells are just like like a middle boundary
Right.
and the upper and the.
So what'll be the chemical field when we run in it? Yeah.
So like, our initial is 1. So I think the bottom part would just end up with 0.
Right.
And here, here just keep accumulating all these.
Well, you have a constant value boundary, so it's actually going to go to the value.
Just end up with like.
Right. If you had constant flux boundary, it would go to infinity. Yeah. That's right. Perfect. So let's do that and see whether that happens. So now we would say diffusion coefficient by cell, and then we'll set that to 0.
Good.
See, now that's why you want to be able to change the diffusion coefficient by cell. Because now you can define regions that have special names. All right, so let's try that. See if that works. Yep.
Yeah.
So now, suppose that would be an impermeable membrane, doesn't let anything through. So we had a situation where we had a totally permeable membrane where it might as well not be there.
Mhm.
Now we have one that blocks everything. Now we can go in between. Now we could say, what if we made the diffusion constant 0.1? So 1/10 of that in the median. And this is why plotting the value as a little plot is helpful, because it's hard to visualize it. But if you have an intermediate diffusion, if the diffusion constant in the membrane is 0, you get something like a step, which is what we saw. If the diffusion constant is the same everywhere, you get a straight line. If the diffusion constant is small but not 0 in the center, you'll get something that looks like that. And that's a little hard to see from the colors by themselves. But why don't you try it just to see?
So you just try only with the plot first, and then like, do you need to try?
We don't need to do the line plot, it's all right.
Okay.
You may have to, that's where those contour lines can help, right? It's because it's a little hard to see. This is not the same, not quite the same as if it were, it's certainly not the same as if it's a complete barrier. What's harder to see is that it's not the same as if it were no barrier at all, that there is this jump across the membrane. Something that's easier to see is when you have a little ion channel. And so now let's overwrite, in the initializer, we're going to overwrite just one tiny square in the middle with medium. And the way you do that
So we just.
is copy this whole region statement, region to region, okay, and then paste it in below that. And most things in the XML are not sequential. This one is sequential. So now if you say, instead of type, you type medium there. And now you pick say X min 46, X max 50. Now see what happens.
Okay.
And actually make the diffusion constant 0 again. Right? It's more fun if you make the diffusion constant 0 in the cell. Now you can see the contour lines, right? The little line.
Mm.
So now you have a little hole in the membrane where you get chemical going through.
That's very interesting.
So that's actually sort of fun. Now, what we can't do with this is active pumping, where the concentration is moved by a pump against the gradient. We can do that by hand, but it takes some work. The other one we can't do is symport, antiport, where you have two chemicals, one of which moves against the other one.
Like the potassium.
Right, they're calcium, potassium, or proton, potassium pumps. Yeah.
Okay.
You can do those things by hand. In other words, if you draw a bunch of cells across the boundary like that, and then you calculate by hand the differences, you can move concentration in Python by hand, say absorb here, secrete here, absorb here, secrete here. But you have to do the calculation. We don't have all of those predefined. The finite volume plugin has some of those capabilities. The diffusion solver, the finite volume diffusion solver, has some of those capabilities. I don't know all of them. I'd have to look at the manual myself to see which ones are available. But we at least have some simple ion channel-like behaviors that you could play with. So that's a sort of a fun thing to do. I want to come back to now decay, because we haven't really talked about decay. To make the decay equation, we add a term which is our diffusion equation, and now we have what's called first order decay. The concentration decreases proportional to the concentration. And we have a factor gamma, which is the decay constant.
Mhm.
And this is going to give us something that's pretty important, which is a concept of a diffusion length. And we can solve the diffusion equation with decay. I'm not going to do that particularly today. But if I just look at the dimensional analysis, if I look at the diffusion constant as units of length squared per time, the decay constant has units of time per time. And so diffusion over decay constant has units of length squared. So the square root of that has units of length. So the square root of the diffusion constant over the decay constant has units of length. And that diffusion length is the single most important thing in biology as far as diffusion goes. Because the diffusion time tells us how long it takes to go a given distance. The diffusion length tells us how far things get. And that really, for my argument about oxygen, I should have used the diffusion length, not the diffusion time. Typical diffusion lengths in tissues, depending on the chemical, are somewhere between 5 and 100 microns. If chemicals decay or if they're taken up. And so this defines the typical length scale of structures of the tissue. If you have the diffusion length right in your simulations, even if everything else in the diffusion is wrong, usually you'll get reasonable answers. And so it's pretty common to having very fast diffusion, like for oxygen, is very expensive computation. If you reduce the diffusion constant of oxygen by 1,000, but you also reduce the decay constant by 1,000, the ratio is the same, and therefore the diffusion length is the same. And provided things don't change very fast in the system, which they don't, nothing changes in your results. Your simulation will run 1,000 times faster, and it'll give you the same result. And so if you are going to pay attention to one thing that's going to be correct about the biology, you want to make sure that the diffusion lengths of your chemical species are correct. And in some sense, this is the single most important thing I can tell you about modeling for spatial modeling. And absolutely with your bacteria exchanging chemical species, this is critical. How far, if you have two bacteria, one is secreting molecule A and one is secreting molecule B, they only know about each other if they're within a diffusion length of each other. And so the size of the clusters of bacterial colonies, for example, are governed by diffusion lengths. So it's absolutely critical to get the diffusion lengths correct when you do simulations. Everything else is negotiable, but diffusion lengths are not. And so, well, the diffusion length determines how far oxygen gets in a tissue, how far glucose gets in a tissue, how far morphogens get during development, what size stem cell niches are, how much cancer is going to be, how rapidly cancer will evolve to be invasive or resistant to therapies. All of those are governed by diffusion lengths. And so it's a very simple number. Now, the problem is that if there's a single decay constant, then this is easy to estimate. In reality, if you have chemical decay and uptake, and the uptake isn't uniform, then what the uptake or decay rate gamma is may be a little bit ambiguous. And so this may not be an exact number. In other words, you may get the diffusion length is 5 microns to 20 microns, but you can always estimate it. You can always estimate it. And it's still going to be important to have that estimate. The other thing is the equilibration time, which is how long the chemical field takes to forget its initial configuration. And that's simply 1 over the decay constant. If there's no decay, then you never lose the initial configuration, which is why we almost always, in a simulation, have some small decay term, because otherwise our initial condition never goes away.
With this, for our previous like uniformization pattern, we didn't set decay. We didn't set
That's
decay
right. We didn't.
factors, but like the ultimate C is totally different from the initial
Right.
condition, so that couldn't be counted as like forgetting about the initial or
Right. Because our boundaries are absorbing boundary conditions. Right. If we had periodic or no flux boundary conditions, we never forget.
Okay. So that's okay.
Think about it this way. Suppose we have periodic or no flux boundary conditions. There's no secretion and no decay. Then the amount of chemical in the system never changes.
So like if a boundary has the capability to absorb or secrete something, that's kind of like
A
decay
decay.
or
Right, it's
decay.
right. The thing is it takes time to get to it. So that's where the diffusion time comes in, because the equilibration time is basically how long it takes for the chemical to get all the way from the middle to the edge, if all the decays happen to get the edge. And so if we had more time, I have a whole bunch of exercises where you plot these things under these different conditions and look at how fast you equilibrate and all of those kind of things. But yes, absorbing boundary conditions behave like a decay. But it's a decay only at the edge, not in the middle. So it takes time for the system to forget.
Sure.
But that's a very good, important insight. Yes, absolutely. So we have a global decay constant. And again, one thing to remember very often is that what you really want to do is, instead of saying, what is my diffusion constant, what's my decay constant, you should start out by asking the question, what is the diffusion length I want in my system? Then pick a value of diffusion constant that's convenient for you, and then pick a decay constant that gives you the correct diffusion length. Again, if you make the system slower, in other words, you make the diffusion constant for oxygen 1/100th of what it really is, and the decay constant 100th of what it really is, then the system is going to take 100 times longer to equilibrate. But once it's equilibrated, the pattern of chemical will be identical. So the spatial pattern at equilibrium doesn't depend on D. It only depends on the diffusion length. That's why I'm saying the diffusion length is primary. Because if the equilibration takes 1 second or a minute, and the time scale in the system is hours, we don't care. So you can speed up the system, the calculation a lot by basically slowing diffusion down. But then you have to remember to rescale the diffusion, the decay constant, and everything else.
Okay.
And so one thing we can do is compare, linear gradient, source here, or decay here, straight line. With decay, you get an exponential curve. And that's typical. Sorry. Let me just quickly, since we don't have a lot of time, I want to talk a little bit about sources and sinks. So CompuCell has, in some ways, a rather flexible language for describing sources and sinks of chemicals. It's better on sources than it is on uptake. We really need some additional uptake rules, but anyway, that's what we have. And so here we're adding removal of the chemical and secretion of the chemical. If we have secretion and decay at the same time, then the equilibrium concentration is the secretion rate divided by the decay rate. And this has got a typo in it. Sorry.
Let's scroll down a little bit.
Yeah.
Previous. Wait, there was after that one.
Oh, because we skipped these slides. That's all. I didn't want to spend a lot of time on the math. So here we are. Sorry. What is that? That's not an equation, it's some,
So you mean like the gamma max or?
it should be gamma, not C.
Okay. Max equals. Oh, I see. So max equals S divided by gamma.
Where did the equation? C equals that must be one very old slide.
That's good.
If you go far enough back, Microsoft had a different equation editor.
Mhm.
They changed the equation editor in Word and PowerPoint. And the current equation editor is not compatible with the ancient one. So if you open a slide that was made 20 years ago, the equations aren't editable anymore because it doesn't support it. They didn't make it backward compatible. So sorry about that. This is such an old slide. It had the wrong.
Things like that usually happen when I was a graduate student.
All right. Anyway, so that's correct. And this is also something that's useful because if you change the decay constant, you have to remember to change the secretion rate to keep the ratio the same. Otherwise, the values will blow up. In reality, diffusion happens according to what's called Fick's law, of course, a membrane, which is where we started with the diffusion equation in the first place, which is the rate of transport is proportional to the difference in concentration.
Mhm.
So if there's no difference in concentration, there's no transport.
Mhm.
Now, in biology, when you have secretion with vesicles,
Mhm.
that's an active transport mechanism. So they can keep pumping something, even if the concentration outside of the cell is higher than inside, the cell can keep pumping stuff out with vesicular transport.
Mhm.
So in general, with vesicles, the rate of transport is independent of the concentration difference. It's constant.
Mhm.
So that's something to be aware of. In CompuCell, secretion is something that we invented 3 times. And so there are 3 different ways of specifying secretion. And they add. So in most cases, if you specify things twice, it either doesn't work or only one works. With secretion in CompuCell, if you specify it in 3 different places, it does it 3 times. You can specify something called a secretion plugin in XML that says cells of a particular type secrete in a particular way. In the diffusion solver, there is lines to specify secretion and absorption of chemicals by cells for that chemical. And as always, we could also specify in Python individual cells secreting and absorbing separately. Now, usually we will wind up having the secretion and absorption handled per cell in the Python. But as always, it's slower to do it that way. It's more flexible, but it's slower. On the other hand, in the situations of secretion and absorption are usually so complicated that we really need to be able to do it. You know, different cells do different things, so we need to be able to do it on a per cell basis. So one way to do it is specify it within the diffusion solvers. there's a block called secretion data. And we could say here, secretion type bacterium, and it's secreting at a constant rate, 0.1, into whatever the field that we called it was. We can say only secrete at the boundaries where this bacterium is touching another bacterium, a macrophage, a wall. And we can have it have a different value from the regular one. So we could say, if the bacterium comes in contact with something else, then it secretes in the direction of the thing that it's in contact with.
It's just by fault.
Sorry?
It's by fault, like the direction.
So if we have secretion on contact, suppose I have secretion on contact between two bacteria.
Mhm.
If the bacteria are all separated, they don't secrete. If they touch, they start secreting.
Mhm.
Or vice versa, if I say secrete on contact with medium and the bacteria are clustered together in a biofilm, only the ones at the surface of the biofilm will secrete. So that's a pretty powerful concept. So you can make secretion conditional on what your neighborhood is. What you can't do is to say secrete in a particular direction. In other words, any surface that's in contact with medium will secrete, not just the top surface. There are ways with compartmental cells, you could make it more direction. You could secrete at a constant rate, or you could secrete at a constant value, constant concentration. And it's convenient sometimes to have constant value rather than constant rate concentration. There is a separate plugin called Secretion, which basically has the same syntax as the one we just did, but it's not inside of the diffusion field.
Is that parallel to like other plugins?
Yeah, that's
Okay.
right. That's right. And again, they both operate. So if you say the cell secretes into chemical field X, it's just the cell secretes into oxygen, you say it with a secretion plugin, and in the diffusion solver, it will secrete twice.
Okay.
We could also define it in Python. And as usual in the Python, we have to specify the plugin. And we just give the field name and nothing else. And it's tricky to remember what has to be in the Python, what has to be in the XML and not. And then in this case, we need to get a reference to the chemical field. We have something called a secretor that basically tells Python how to secrete into that chemical. That basically is a handle for secretion. And then we have to tell the CC3D per cell what to do. And there are a bunch of options. We can secrete inside the cell at the boundary, inside the cell at the boundary on contact, at the center of the cell, everywhere in the cell, just outside the cell at the boundary, just outside the cell at the boundary contact. And we can also ask it to tell us how much it's secreted at each time step. And for secretion inside cell, we can specify constant value rather than constant flux. Some of these don't make any sense for constant value. And so you have something that looks like secretion plugin with nothing else. In the Python, we have get field secretor, name of the field. And then we need that to do things that we're going to do. And then we would say things like, we get the field secretor, and then attractor secretor total field integral tells us the total about in the chemical. That's equivalent to saying self.field.chemical.max. I'm sorry, this is total. Sorry, there's a total one also the other way.
Okay.
If I want to know how much is available to the cell, there's amount seen by cell. That gives you the total amount of field in the pixels occupied by the cell. So we can read a lot of information about the chemical field on a per cell basis. There are some things you want to do that wind up not being available because there are an awful lot of possible combinations, and we have a lot of them, but not every single one. So sometimes you don't get it. And so you could say attract or secrete at inside cell at boundary, attract or secrete inside cell total count, tells me how much there was. Amount secreted, which is returned by attractor, is an object itself. So you have to, it's not, this returns total count doesn't return a number, it returns an object. Total count dot total underscore amount is the number. We'll come back to that next week. I mean, I just want to show you a few things so you've seen it, and then we're not going to do it. And again, here are the options. Here's secretion inside cell at boundary. Here's secretion inside cell on contact with medium.
So,
The red is showing where it secretes.
okay, so it's like inside the cell.
It's the pixels just inside the cell. Pixels just at the surface.
Right picture, just like with an additional condition.
Right, yeah.
Okay.
And here's just outside the cell, which is maybe more realistic on contact with medium. So if the cells were secreting into the medium, the chemical would show up just outside the cell, right? So that's what that does. And there could be total counts. So there are a lot of permutations here. The function names get pretty long because we're trying to be descriptive. Secrete outside cell at boundary on contact, total count. It's a long list. But it's nice that you could do it. If you're imagining something in 2D where you're secreting everywhere on the surface, then secreting over the whole cell might make sense. To support things like physicell that are point particle things, we let you secrete or absorb at the center of the cell.
I'm sorry, like center? Like,
Physicell, because it's a point-based model, it can't secrete over the whole volume the way we do. So if you want to match a physicell simulation, you could say, just put the chemical at the center.
it would not like fill the volume.
Well, then it'll diffuse through the volume, but the point is, I don't recommend doing this, but it's there because when people want to compare center models to CompuCell, it's useful to have that.
Okay. I have another question.
Yeah.
So what is the 300 here? Like,
I just picked a rate. That's some number, the rate of secretion.
Oh, okay.
That's 300 units of chemical per time step.
Oh, okay.
Again, CompuCell is not smart about units. So if you change the length scale or the concentration scale or the time scale, you have to rescale it by hand.
Okay.
Which is a pain. You should have a constant in the beginning of your Python, which is length unit, time unit, concentration unit. And then you should write secretion rate is alpha times length divided by time. So you should write it, you should have it scaled. You should write it scaled, so if you change the length and time scales, they scale automatically.
I think I'm beginning to like have a deeper feeling about this. Just like in last week's assignment, like I'm required to do some constraints in like the shape. So you have to combine, or you have to tag approximum based on other. Yeah.
I mean, some of the slides I skipped have the mathematics of how to get those correction factors. But I mean, it's arithmetic, it's not complicated, but it's tedious to do that computation. If you're just trying to do a toy model, or you're not going to be changing the resolution of your simulation, then it doesn't matter. But if you want to write a flexible simulation where you could change the length scale of the simulation or the time scale, then it's worth taking the time at the beginning to write in the scaling factors. If I were going to do CompuCell again, I would make it so that automatically we have those things scaled by the length and the time properly. There's a paper by Murray, Stan Murray, where he calculates all the corrections that are needed. So if you don't want to calculate it, but you don't want to work out these days, you could probably ask ChatGPT, what is the correction factor for this parameter? And it'll look at the units and say, okay, you need to multiply by the length scale and divide by the time scale, whatever the corrections are. So it's easy to make a mistake and get it wrong, but it's possible, but it is something just to remember. So the reason I picked 300 was specifically so that it's something that doesn't look reasonable, so people remember they have to change it. All right. And then I have some exercises where you have secretion. And again, depending on the rules, here you have decay, so you get, here's a Gaussian concentration field. Here's a constant concentration, so the field is constant at the top. And so here are all the different kinds of secretion, and you get here's the curves for each one of them. They're all slightly different. If you go far away from the source, they're all the same. Distance, they're all the same, but near the source, they have different profiles. Uptake is a little more complicated. I can't uptake, I can secrete at a constant rate. I can't take up at a constant rate, because if I do that, my concentration can go negative. I can't take chemical out of the system if there isn't any. And so usually uptake is done as if it were decay with first order kinetics. Now, really, we should have more complicated uptake functions, because in reality, the uptake is often Michaelis shaped or Hill function shaped or something complicated. CompuCell only lets you do a saturated uptake. So CompuCell allows you to say, take up a given percentage of the chemical up to a maximum amount, and then take a constant amount after that. So if we had a Hill function, which was saturated, now this would be the half max, and then this would be the max value. And this is the best we have. So here's uptake inside cell, at boundary, total count. How much did we take up at the boundary inside the cell? Max amount is the saturation level. Relative amount is the slope. And that's what we use most of the time. And so that's and then you have all the different configurations. And I think let's stop for today. That at least you've seen the commands now. Do you have anything you want to talk about or questions for today?
Okay, I have a very simple question.
Yeah.
Technically, so when I was trying to adjust the lambda surface for lambda volume, and I noticed they both appear in stackable high and XML. So when I was trying to change them, so I should change all of them, where I only need to change the value in the stackable file.
Okay, so if you specify lambda volume in the XML, you cannot change it in the Python.
Uptake. So that means like, even though I write comments that cell dot cell dot lambda value with cell dot lambda surface, and like that equals plus something.
In the
But
Python, it will do nothing if you specified it in the XML.
okay, but like, but why? Like when I was trying to let the cells grow, like
Right.
why they work? Because I changed the target volume. Oh, that's the target volume. Sorry.
No, but target volume you can't change either. If you specify it in the XML, you cannot change it in Python.
But so why does the grow in the growth model that changed? Like this is the kind of
Can you show me? Because
but like you
unless the code, unless somebody changed CompuCell. So maybe they fixed it. This has been a bug in CompuCell for 10 years, 15 years. If you specify the target volume or the target surface in XML, you cannot change that value in Python.
you mean that's for the initial condition, or because like in growth model we would say the cell.
You want to screen share and show me what you got, what you're saying? Because I'm happy to see it, but I unless I see it, I don't think I understand.
Oh, okay.
I think I know what you're saying, but I'm not 100% sure. So why don't you show?
Okay. Findings. Project.
I mean, I would have agreed with you that the XML should specify the initial condition and then you can override it in Python. But it doesn't work like that for the volume and the service constraint.
Just overall see if the code is one. Okay. Like for example,
You want to screen share it? Now I can make it
Okay.
just easier to see.
yeah. So for example, here, like I make change to the cell target volume. So that can be kind of.
Wait a sec, hold on a sec.
Okay.
I don't know why it's not, okay, okay, so let's see, so. Maybe make it a little bigger, because it's hard.
Oh, it's kind of like the main. It's kind of like it.
Yeah, I don't know why it's so slow, yeah? Let me. Okay, so let's look at your XML. Okay. So look at line. So look at line, what is it, 20, 30? I can't see the numbers. 29, 30, 31, I think. 31. Yes. You see, you have just volume there. You don't have target volume and lambda volume specified.
Like, okay, can I try to find another model project?
I mean, what you have there is correct and it will work.
Okay.
I mean, we could do it the other way, which is if we put lambda volume and target volume in, I can show you it won't work.
Okay. Let me try to find existing one. So. Oh. Oh, see, like here, this is somewhere like sat along the surface along the value.
Yeah. So now if you go to the Python, if you try to change those, it won't do anything.
Like here,
That won't do anything.
oh, that's why even though I write the target surface needs to like have this kind of relations with the target volume, but the cell still appears as
Right. So good. So run that simulation.
cycle.
So when you run that simulation, the cell will not grow, right? Doesn't grow.
Yeah,
No growth.
yeah, and they like
Okay, so now let's change. Let's go to the XML.
mhm.
Go to the line. Go to the XML. And we need to find the XML. And now comment out the line that's that line that you've got right there.
Okay.
And you can do that with the, okay, you can delete it too. And you have to do that one. Okay, good. Now save it
Let's just say
and now run it.
Oh, okay. 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 I think I think a lot of my homework.
Okay,
It's
so I don't like to say I told you so because this is a problem. I make the same mistake. But I've said every class so far, I've warned you that if you specify these things in the XML, you cannot overwrite them in the Python.
okay.
Every single class I've said, be careful if you specify them in the XML, you can't overwrite them with the Python. And even though I teach it, I make the same mistake. So I promise you, I believe that the XML should be the initial value, and then you should be able to overwrite it in the Python. But this is something that's very deep in CompuCell. And it's because originally we only had the XML. The Python was added later. And the way the C++ exposes the volume and surface constraints, it doesn't allow, if you specify it in XML, it doesn't allow you to do that. Now, so I apologize for that, but that's what that is. The other thing is if you specify the volume or the surface in XML, cell.pressure doesn't mean anything. The attribute cell.pressure will exist, but it won't have a meaningful value. Cell.pressure only returns a meaningful value if you specify them in Python.
Okay. So because
You can still calculate the pressure by hand as 2 times lambda volume times volume, actual volume minus target volume. But you have to calculate it by hand. The attribute cell.pressure does not return a meaningful value if you specified the volume in Python.
I see.
If we had more money and more people to work on CompuCell code, we would fix these things.
I think.
If I had somebody who was really, full-time fixing things, those are things that we would fix. But that's a common problem, unfortunately.
I think that's a lesson until you really like, not some problem. Can you like realize, like what is trying to say?
Well, that's right. That's why I was saying that sometimes I'll do, I'll do as part of the homework, I'll give you a little homework problem on something we haven't played with yet. Not to be mean, but instead, because that way you're ready to say, why is he telling me this thing? I mean, this